Hello, everyone. Welcome to this session. My name is Hossein Sharifi Noghabi. I'm with the School of Computing Science at Simon Fraser University. It is my pleasure to present our work, AITL, Adversarial Inductive Transfer Learning with Input and Output Space Adaptation for Pharmacogenomics. I would like to emphasize that this was a team effort of Shuman Peng, Olga Zolotareva, Colin Collins, and Martin Esther, as well as our generous supporters. Imagine you have a data set of very nice drawings, and your goal is to classify them into these classes that you see. The problem is that you don't have enough samples, and you're a graduate student. You can't afford to hire professional artists to draw more images for you. What would you do? Well, one solution is that to draw them yourself. But if you're a fake artist like me, it's not going to happen because you can see the difference between the nice drawings and what you did. The other solution is that you can go online and look them up over the internet. And it's good because now you can have enough image for each class. But if you train your model using the images that you found over the internet and test it on the nice drawings, your model is not going to generalize well because your train and test data are coming from different distributions. You're training your model hoping to target here, but instead you're going to miss the actual target. You may get some hit, but it's not why you trained the model in the first place. Transfer learning attempts to solve this problem by leveraging the knowledge in all of the available resources that you can get in one learning system. In traditional machine learning, we have separate learning system for each task and the domain associated with that task. But in transfer learning, we have only one learning system, and we want to use all of the available resources to achieve a better goal on the task of interest or the target task. Transfer learning can be categorized into three categories: unsupervised, transductive, and inductive. Unsupervised, as the name indicates, we don't have any label in neither the source nor the target, and domains are different. In transductive transfer learning, also known as domain adaptation, we only have the label in the source domain, and the target is unlabeled. But we have similar tasks, meaning that if we want to classify to 10 classes, we have similar classes for the target domain. And the important thing is that domains are different. Back to our drawing example, this is exactly the case of images that you found over the internet and the nice drawings. And the last category, inductive transfer learning, we have labeled in the target domain. Our source can be labeled or unlabeled, but now we have different tasks, and domain can be the same or different. Moving to driving application of this paper, in drug response prediction, ideally we have a group of patients, we get some omics data type, we can treat them with one or two drugs, build a computational model, and hope that model generalizes to future patients. But patient data sets with drug response are often small or not publicly available. And you cannot treat a patient with multiple drugs separately or in combination. It's not possible. To solve these problems, researchers have focused on preclinical resources, such as cancer cell lines or animal models. The idea is the same. We get some omics data type. Now we can treat them with different drugs separately or in combination to study the effect. We build our computational model and hope that that model generalizes to patients. But there are some challenges in drug response prediction. We have different sort of inconsistencies and discrepancies between clinical and preclinical resources. For example, gene expression as an input data is different between patients and cell lines because cell lines are a bunch of cancer cells on a plastic dish, and there is no, for example, immune system or tumor microenvironment. Also, measure of the drug response is also different between cell lines and patients because in cell lines we have continuous measures such as IC50, but in patients we have categorical measures based on change in the size of tumor. There are also studies that show that even two cell line data sets can have discrepancies. How can we achieve accurate prediction with all these discrepancies? Well, you have two options. You can either train your model on preclinical data sets and validate it on preclinical data sets too. This way, you are minimizing the risk of discrepancies between your train and test data but you don't know about translation to patient. The other option is that you train your model on preclinical data and validate it on clinical data. And this way you can study translation to patients, but you need to find a way to address the discrepancies. Two general ways to address discrepancies between pharmacogenomics data sets is that you can either obtain data and integrate the data set and try to standardize them and homogenize them in a same way, essentially pre-process and prepare data across different resources similar to each other. Or your other option is that design your predictor in a way that can handle discrepancies in input data. In this case, this paper aligned principal components between cell lines and patients to achieve accurate prediction on patients. But in drug response prediction, we have both discrepancies in input space and the output space, because as I said, gene expression data obtained from patients is different than gene expression obtained from cell lines. And measures of the drug response are also different between cell lines and patients, which means that this drug response prediction falls into inductive transfer learning category. And there is no method available to address these discrepancies at the same time. And our goal is to design the first method to achieve this. So before moving to our method, first I need to present some of the related works to make sure that we are all on the same page. These related works are on multitask learning, generative adversarial networks, and adversarial adaptation. The idea of multitask learning is that traditionally we have learning systems with one task, but now when we have multiple related tasks, we can train them jointly in one model. This is especially helpful when you have limited samples for some or all of these tasks. Because of the sharing parameters that happen at initial layers, the training is more efficient in multitask learning. In a very common and simple scenario, for example, you can have a couple of shared layers and a couple of task-specific towers for the tasks that you are training the model for. And we will use this in our method. The idea of generative adversarial networks is that we have two competing networks, a generator and a discriminator. Generator receives nothing but random noise and creates a fake image. The discriminator receives that fake image and some real image from the training set and should be able to discriminate these inputs into real and fake. And in this min-max training framework, generator eventually learns the distribution of the input data. The generator wants to minimize its error, and the discriminator wants to maximize its accuracy of capturing real and fake. And a lot of people use this clever idea in the concept of domain adaptation using adversarial adaptation. In this example, this paper ADA, they had a source and target images. We don't have the label for the target domain. In the pre-training step, they use the source images to train a feature extractor, a standard convolutional neural network, and a classifier to predict class labels. In the adversarial adaptation step in the middle panel, they kept that feature extractor for the source domain frozen. They did not fine-tune or retrain that, but they trained a target CNN and a discriminator in an adversarial way, meaning that the extracted features from using the target feature extractor and source feature extractor should be domain invariant to fool this discriminator. When we have such a feature extractor for target domain that generates features similar to the source domain, we can use the train classifier on source examples that we had in the pre-training phase to make prediction for the target examples. With this quick background, I'll move now to our method, AITL, Adversarial Inductive Transfer Learning. Our method takes gene expression data from cell lines and patients as source and target. Remember that we have continuous measures of the drug response for the source domain, for the cell lines, and we have a binary measure of the drug response for patients, for the target. These are the input for our method, and we use a feature extractor to learn high-level features in lower dimension. We denoted the feature extractor by F and the extracted features by Z. After extracting features, we want to make these features domain invariant by addressing the discrepancies in both the input and the output spaces. To addressing the discrepancy in the output space, we design a multitask subnetwork that takes the extracted features using a shared layer denoted by G and two task-specific towers denoted by MS and MT. MS has a regression loss, and MT has a standard binary cross entropy loss. This is because we have the continuous measures of the drug response for the source, which means that it's a regression problem, and we have binary measures of the drug response for the target, which means that it's a classification problem. And this sharing parameters also tackles the limited number of target examples that we have in drug response prediction. So by training this multitask subnetwork, we can solve the output space discrepancy because the classification tower that makes prediction for this patients can also make predictions for the cell lines, which means that we can assign cross-domain binary labels to the source samples, because we didn't have binary labels or categorical labels for the source examples. And with these labels available, now the output space discrepancy is resolved. To solve the input space discrepancy, we use the idea of adversarial learning by designing global and class-wise discriminators. A global discriminator receives the extracted features from source and target and should be able to discriminate them accurately. And the idea is that we want the extracted features from these two domains be so close to each other, be domain invariant to fool this discriminator. Now that we have binary labels available, thanks to the multitask subnetwork for the source domain, we can put further regularization on learning domain invariant representation by having class-wise discriminators. A class-wise discriminator receives the features, extracted features from source and target for a specific class. For example, if it's class 1, we only input samples of the target that were associated to that class, and those of the source domain that we predicted as class 1. And the whole network is optimized end-to-end using the following objective functions that you see on the bottom. And we have binary cross entropy for classification, mean squared error for the regression, adversarial adaptation loss for global discriminator, and adversarial adaptation loss for class-wise discriminators. And we use the idea of gradient reversal layer to train this network end-to-end. We are seeking to answer 3 questions in this study. Can we outperform the method of pharmacogenomics that they don't do any adaptation? Basically, they're trained on cell lines and validated on patients. Can we outperform those methods that can address only the input space discrepancy? And if we use our method to make predictions for patients in TCGA without any drug response, if those predictions mean anything? To answer these questions, we use the following resources for training and validation. As the source domain, we use a panel of 1,000 cell lines from GDSC dataset. These cell lines are screened with 265 drugs. All of the expression data are from are obtained pre-treatment, and obviously the response outcome is post-treatment. And for the patient data set, we use a clinical trial and preclinical data sets from the following resources. We use a 3-fold cross validation on patient data set, meaning that we use 2 folds for train and validation and the 3rd one for test. And similarly for the train set, we had 3-fold cross validation on that one as well, 2 folds for train and the 3rd fold only for validation. We didn't use cell lines for the test. And we had TCGA also for independent tests to study association. And based on availability of enough number of samples, we focus on the 4 drugs that you see on the right side. When we apply these data sets to our method, here you see our first evaluation on area on the ROC curve. Let me walk you through this table. The first 2 methods, they don't do any adaptation, and they were specifically designed for pharmacogenomics. Method of Gleher is a regression-based pipeline, and MOLI is a deep neural network. The next 3 methods, Precise was specifically designed for pharmacogenomics. The other 2 were designed for computer vision applications that we adapted them for this problem. and they only do input space adaptation. And protonet is a method of few-shot learning that adapts the output space. The second part is the adaptation study, where we remove different components of our method to study the impact of each module or subnetwork separately. And the last part is our method in its complete format. And as you can see, our method, using all of its components, consistently outperformed our baselines, which means that addressing the discrepancy in both the input and output spaces improves the drug response prediction, in this case, significantly. We obtained similar results in terms of precision and recall, and our method, the last bar in dark green, outperforms the baselines across the studied drugs. And that horizontal red line is random performance for precision and recall. To study the impact on TCGA patients without any drug response, we followed the evaluation setting used in previous works. In this case, we use the trained AITL for different drugs on some of the TCGA cohorts separately. Then we selected known target genes from pharmacodb resource, and we fit a simple linear model between the level of expression of those genes and the response that our model predicted. And as we expected, we found significant associations between the known annotated target genes and the response that our model predicted across different cancer types, which means that the response that AITL predicted, they were not random, and they actually meant something with respect to the target of the studied drugs. So in summary, we proposed the first method of inductive transfer learning. For pharmacogenomics, our method is the first method that can adapt both input and output spaces. And we showed that our method using multitask learning and adversarial learning can significantly outperform a state-of-the-art of pharmacogenomics and transfer learning. For future work, we acknowledge that AITL needs patient data with drug response as an input for training, which means that we need to deal with this limited sample size and other problems that I mentioned associated with patient data. And the question is, can we use alternative large resources such as TCGA without any drug response and leave patient data sets with drug response only for independent tests? And this means that our method or future work, they have to use these data sets as an unlabeled data set because we don't have any drug response in resources such as TCGA. And finally, I would like to thank all of you for joining this session, and please do not hesitate to contact me or my collaborators if you have any questions. All the codes, data, and required materials to reproduce our results are available on our GitHub. Thank you.